Hello students, today we will learn about biotechnology and its applications. This is chapter number 12 in 12th class biology book. In this video we will learn about the different areas in which we can use biotechnology to get its benefits like agriculture, medicinal industry, in therapeutics, in diagnostic techniques, in molecular diagnosis, in bioremediation we use biotechnology. And these are few areas where we use biotechnology. Let's start it. Biotechnology. Biotechnology is a science in which we use living beings and apply technology on them. We modify their DNA, we change their DNA, we get the desired gene or the desired product from that DNA. It deals with industrial scale production of biopharmaceuticals, biologically using genetically modified organisms, GMO, that is the genetically modified organisms, which can be microbes, which may be the fungi, plants and animals, and these microbes are used as models to study the different kinds of diseases, their pattern of inheritance, their effects, their phenotypic expression and so on, and the drugs, how the drugs treat particular disease. These all areas come under the biotechnology. Its application include therapeutics. जैसे कि हमने किसी चीज़ की थेरेपी देनी है, थेरेपी मीन्स ट्रीटमेंट, तो ट्रीटमेंट में बेटा हम बायोटेक्नोलॉजी को यूज़ करेंगे. फॉर डायग्नोस्टिक, इफ़ वी हैव टू फाइंड आउट द kind of disease the person is suffering from, we use biotechnology in certain cases like ELISA, PCR are few techniques which are used for molecular diagnosis. Then we use, for improving the agriculture produce, we use genetically modified crops, we produce processed food, we do bioremediation. For the wastewater treatment we use bioremediation for wastewater treatment as well as for energy production. These all areas use biotechnology. This slide is about the different areas where biotechnology can be applied. Traditional genetics. Traditional genetics is the simple hybridization method which we used earlier, that is selection of the good variety of the plants or animals, then make a hybrid plant or animal. This is the traditional method which we use. And then we started using biotechnology, we started mutational breeding and then select the particular organism. Like firstly we induce the mutations and then we select whether these induced mutations are beneficial for us and or not, and then we use it and apply it for in different areas. Like mutant plants can be obtained, antibiotics can be obtained. In molecular biology we use biotechnology like recombinant DNA technology, recombinant proteins बना लेना बेटा, genetically modified organisms, gene therapy, नई antibiotics बनाना या कुछ नए molecules बनाना, these all come under the category of molecular biology. Then sequencing technology, which is used in forensic sciences for forensic detections. crime spot पे कोई samples मिले हैं, वहाँ से DNA extract करना, DNA fingerprinting करना और उनको suspects को जो जिन पे हमें suspect हैं हमारे लिए कुछ, उनमें से find out करना कि who is the criminal. In this way we apply biotechnology in forensic detections, and we also use biotechnology in gene banks. Human genome project आपने बेटे पढ़ा है previous slides में. There also we use biotechnology, and then to find out the evolutionary relationship between the different kind of organisms. Then it also deals with the DNA synthesis. DNA synthesis, that is detection of probe, artificial gene making and gene mapping. These all categories come under the category of DNA synthesis, which are part of molecular biology. Then controlling the gene expression by using the RNA interference method. The next field where biotechnology can be applied is cell culturing, like cloning of organisms and stem cell technology and monoclonal antibody technology. By using monoclonal antibodies we produce anti-cancerous drug. These monoclonal antibodies are effective against detecting the particular kind of antigen find in the cancer patients, or whether the oncogenes found in the cancer patients. Monoclonal antibodies are also used for making the anti-cancerous drugs in diagnostic and in nanotechnology. Then the other field of biotechnology is bioinformatics, and the bioinformatics has these further areas, that is genomics, which deal with the study of all kind of genes, genetic disorders and their inheritance from the parents to the offspring. Then individualized therapy which should be given to treat the genes. Then it deal with proteomics, that protein का design क्या है, एक particular gene जब express होता है तो that get expressed in the form of protein, तो उस पर्टिकुलर जीन से कैसा प्रोटीन बनेगा, उस प्रोटीन की क्या 3D स्ट्रक्चर होगी, उस प्रोटीन को कैसे मॉडिफाई किया जा सकता है. अगर उस किसी पेशेंट को हम कोई ड्रग देते हैं तो वो जीन के बने हुए प्रोटीन के साथ कैसे इंट्रैक्ट करती है, उसके बेसिस पे ड्रग डिज़ाइनिंग करना, दिस ऑल कम अंडर द कैटेगरी ऑफ प्रोटियोमिक्स. देन माइक्रोएरे दैट डील विद द ड्रग डिज़ाइनिंग एंड द जीन एक्सप्रेशन. Then computer aided drug design, these all come under the category of bioinformatics. Now, three critical research areas of biotechnology are: first, it provides best catalyst in the form of improved organism. हमने यहाँ जो बायोटेक्नोलॉजी में जो ऑर्गेनिज्म हम यूज़ कर रहे हैं, वेदर इट इज़ माइक्रोब और अ प्लांट सेल और अ एनिमल सेल, हमने वो जो मॉडिफाई किया है दैट शुड बी प्रोवाइडेड. हमने खुद उसमें मॉडिफिकेशन की है, रिकॉम्बिनेंट डीएनए डाला है, तो दैट रिजल्ट इन इम्प्रूव्ड ऑर्गेनिज्म, चाहे वो माइक्रोब है, चाहे वो प्लांट है, चाहे वो एनिमल है. देन क्रिएटिंग द ऑप्टिमल कंडीशन थ्रू इंजीनियरिंग फॉर कैटलिस्ट टू एक्ट. हम बायोटेक्नोलॉजी में ऑप्टिमम कंडीशंस का ध्यान रखेंगे कि पीएच, टेम्परेचर, ये सब रेगुलेटेड वे में हम ये सारी टेक्निक्स जो अप्लाई करते हैं, ऑप्टिमम कंडीशंस मेंटेन करते हैं ताकि कैटलिस्ट प्रॉपरली एक्ट कर पाए, ताकि रिकॉम्बिनेंट प्रॉपरली एक्ट कर पाए या अपने आप को एक्सप्रेस कर पाए. देन वी डू द डाउनस्ट्रीम प्रोसेसिंग. डाउनस्ट्रीम प्रोसेसिंग में हम क्या करते हैं, जो हमने रिकॉम्बिनेंट जीन या रिकॉम्बिनेंट प्रोटीन बनाया है, उस प्रोटीन को प्योरीफाई करना, उसको मॉडिफाई करना और उसके ट्रायल्स, ह्यूमन ट्रायल्स कर लेना, टेस्टिंग कर लेना, देन उसे मार्केटेबल बनाकर उसको मार्केट में लॉन्च करना. दैट ऑल कम अंडर द कैटेगरी ऑफ डाउनस्ट्रीम प्रोसेसिंग. तो दीज आर द थ्री क्रिटिकल रिसर्च एरियाज which must be considered, and the care must be taken that these should be followed. These three parameters should be satisfied while using biotechnological methods. Now, the biotechnology is also used to study the inherited diseases, and it also help in development of vaccines. These are few branches like medical biotechnology, which is a branch of biotechnology in which we can use DNA fingerprinting to solve the paternity dispute. We can do gene profiling, we can do prenatal diagnosis of inherited diseases. प्रीनेटल डायग्नोसिस का मतलब है कि अभी जो बेबी है वो डेवलप हो रहा है मदर वूम के अंदर, एम्ब्रियोनिक स्टेजेस में है, तो उसको कोई जेनेटिक डिसऑर्डर होने के कितने परसेंट चांसेस हैं, इनहेरिटेड डिजीज़ होने के कितने परसेंट चांसेस है, वो हम सब डायग्नोज कर सकते हैं बाय यूजिंग मेडिकल बायोटेक्नोलॉजी. वी कैन यूज़ फॉर फार्मेको genomics, we can use it for skin grafting, for developing vaccines, then for biopharmaceuticals like for production of insulin, which is used to treat diabetic patient, somatostatin, which is growth hormone, then interferon production can be done, then gene therapy can be given in medical biotechnology. These are certain areas where we can apply medical biotechnology. This pie chart is explaining the different areas the biotechnology deal with, or we use biotechnology in these areas, and the percentage given role played by biotechnology in these areas. Like in pharmaceuticals, we use biotechnology, and 20% of biotechniques are used in pharmaceutical industries, 10% in regulatory industries, 10% in forensic sciences, 15% in food industry, 15% of environmental issues are dealt with the help of biotechnology. Then biofuel production done by using biotechnology is again 15%. And medical lab, where we use PCR, ELISA and certain other biotechnological techniques, we use gene therapy, and its contribution is again 15%. Now let's recapitulate that what we have learned today. We have learned about biotechnology, which is the technology which deal with the modification of living beings by modifying their genes or by modifying the expression of genes to get the desired product. And the different research areas of biotechnology like pharmaceutical, agriculture, medicinal area, then bioremediation, environmental issues which are dealt by using the biotechnology, bioinformatics, medical biotechnology is there, and gene therapy is there. So these are certain research areas with which biotechnology deal. In food industry we use biotechnology for making the good quality and quantity of food. So these are certain areas. In next video we will do the different areas like in agriculture, how biotechnology is playing important role. So that we will do in the next slide. I hope you enjoyed this. That's all for today. Thank you.